We can speculate, we have evidence, and I'm going to present some of that here in this slide. But the reality is we don't know how it happened. We think we have a pretty good handle on when it happened, and that's here. Over 3.8 billion years ago, there are isotopic remnants in rocks. And that's shown here. This is a study that came out decades ago now. There have been follow-up works that have confirmed this, though. If you go to some parts of the world, in Greenland and Australia, there's actually really old rocks. So much of the Earth, because of plate tectonics, has been subducted and taken down back into the mantle and melted and metamorphosed and all those things that you've learned if you took a general geology course. Um, but so most of the Earth's surface is actually relatively young, and that's how we end up with places like California, where you've got plates crashing together and mountains forming and earthquakes happening and volcanoes forming. But in some really remote parts of Central Australia and in parts of Greenland, that ground has never been subducted. And so these make really good places to go find really old rocks. And so that's where they've studied these 3 billion plus year old rocks. And it turns out that if you grind up those rocks and you look at the isotopes in them, now these are not radioisotopes like you've learned about in terms of carbon dating and things like that. These are stable isotopes. But, and so, for example, this is the stable carbon isotope C13, right? The normal isotope of carbon is C12. But C13, the heavier isotope, is found in fairly small amounts, okay, all the time, right? So part of carbon on our planet right now is in C12. Most of it is C12, but some of it is C14, which decays. We're ignoring that for now. And some of it is C13, okay? Now, it turns out that life has a bias towards C12. It prefers the lighter isotope. And so if you see a change or a skew in the ratio of carbon 12 to carbon 13, you can use that as an indicator of life. And that's essentially what they've done in this. So you don't need to know all the details here, but basically, if you see a change in away from inorganic carbon, okay, where you wouldn't have a skew, and if you see lower values here, that means it's depleted in the C13. It has more of the C12. And this is an indicator that biology was going on instead of just geology. Okay. So that is pretty good evidence. And as I said, it's been confirmed in subsequent studies as well. There's also very old fossils. Now, these are somewhat controversial because some people don't agree that they're actually fossils. They have organic carbon in them, but are they actually fossil microbes? That's the challenge. But there's a former researcher at UCLA, William Schopf, who was very famous in the 90s for coming up with this hypothesis. He says that these images, these are thin section EM images, and we can identify that this is filled with organic carbon, fossilized organic carbon, which we call kerogen. And what he saw when he looked at this was something like this. And this is very similar looking to modern cyanobacterial filaments. So he actually said that these were fossilized cyanobacteria from 3.5 billion years ago. Now, there's many folks that don't agree, and there's really no evidence that it was an autotroph. And we think cyanobacteria probably actually came later, but something needed to make those fossils. And so if it was a microbe, it could have been a chemoautotroph or a heterotroph or something like that too. That would be really interesting. The other thing it would do is it would suggest that you have fairly complex life, right? Like, so if we look at this picture, right, we would not imagine the very first bacterium or archaean or proto-prokaryote would have a filament, a structure like this. So this suggests that there may have been fairly rapid evolution, biological evolution in the first half to 1 gigaannum, which is billion years. And then, because things look fairly similar to that from then to now, he argued that we've had slower evolution since then. I think again, this is fairly controversial, but the fact that there are these fossilized remains of things that look like what we see as modern bacteria today is quite interesting. We actually see more complex structures as well. So what this is called a stromatolite. The geologists identified these long ago as of biological origins. They're filled with organic chemistry, organic chemicals like fixed carbon. And what you can see here is that they often have these kind of humps or domes, but can you guys see that there's layers to it? That's the key. So it turns out that if you go to a few places on the Earth today, you can still see modern stromatolites. The reason these are preserved in the fossil record is that they are made of calcium carbonate. What do you guys know that's found on the Earth today that is found in the oceans and is photosynthetic and has calcium carbonate structures?
Diatoms?
So diatoms actually have silicon dioxide, and that is preserved in the fossil record. So what we're looking for is calcium carbonate. So there's microbes. Here's a hint. It's a symbiotic association between microbes and animals, and it's photosynthetic.
Coral reefs?
Yes. So what we think actually are that these fossilized stromatolites may have occupied the niche that's currently filled by coral reefs. So these would have been structures that formed in shallow fringes. And if you look at that Scientific American slide, you'll see they have pictures of little stromatolites in the shallows. So it needs to be shallow because you need to be able to see the sun. But we know that on modern shallow seas in places like Northern Australia and in the Bahamas, we can find calcium carbonate containing stromatolites that are dominated by cyanobacteria. So they're carbon fixers, okay? But those cyanobacteria are often found with very diverse communities of other heterotrophic organisms and even other autotrophs that live there. And so that's actually, I have studied a number of non-calcifying or non-hard microbial stromatolite type structures in the microbial mats that we study. But the ones that I study are soft, and so they would never be preserved in the fossil record like these stromatolites are. Okay. So this just gives us kind of a general viewpoint. This is taken from now a quite old edition of Brock's Biology of Microorganisms. But what it kind of gives you is a sense of when the timing is, right? And the thing we need to think about here is that, we're up here, right? We're not even on this, right? Because dinosaurs don't come in until here. And we think of dinosaurs as being, hundreds of, tens to hundreds of millions of years ago, right? All of this that occurred would have occurred during what we call the Precambrian, when most of evolution occurred. Okay, early origins of life, over 500 million years ago. So animals and modern plants and all of those are way up here. But what you can see is that it took potentially billions of years, but around 3.8 billion years ago, we think life originated due to those isotopic measurements and some of those fossils. It has since differentiated into the eukaryotes, the archaea, and the bacteria. We know cyanobacteria must have originated by probably 2.8 billion years ago, and that's because, as you can see on this right-hand side, it went from anoxic to oxic, and we can see this in the fossil record as well. Iron in the sediments oxidized, and that's a good indicator there's oxygen in the atmosphere. So what you can see is that oxygen levels have been going up and up and up, and that's because we have now green plants and we have lots of photosynthesizers. So, eukaryotes may not have evolved to between 1 and 2 billion years ago, but still remember this is so long ago. Okay, we don't know what this is, when exactly this happened, but it's an important concept for us to think about. All right. How this happened, how we ended up with the exact origins of life, we don't know. As scientists, we're looking for, you know, evidence-based kind of things and not dealing in sort of theological explanations. But the reality is, there's good evidence that it did occur here. So how did this happen? Well, there's some speculations, but really much of what that is just that. So take it for what it's worth. In the 20s, there were some researchers, Oparin and Haldane, that came up with this idea. Given the lack of oxygen and so-called reducing conditions there, and then there would have been UV radiation and lightning. So they came up with kind of the prebiotic soup evolutionary model, and that's what many people sort of refer to. Okay. This was followed up in the 50s by some seminal work out of UCLA. Stanley Miller and his postdoc, Yuri, did some experiments to try and show that you could come up with complex organic materials without life, right? And so they tried to recreate what conditions might have been like at that point, and then they zapped it with electricity to mimic lightning, right? And they found they never formed DNA, they never formed life, but they formed fatty acids and amino acids. Now, one of the reasons that they weren't successful in generating life in a test tube, there's probably lots of reasons, but one of them is they were wrong about the conditions. We're still evolving in our sort of understanding of what the early Earth's atmosphere was like. And so we've never been able to sort of bridge this gap. That said, these experiments are very interesting in determining how sort of organic materials can be synthesized, et cetera. Now, we mentioned that it was really hot, and that's indeed what we think was going on. And so it's interesting to think about what organisms might have lived very early on, and we can see that they would likely have been thermophilic, meaning heat-loving. And this is actually supported by some evidence. Now, I know this is a little different than maybe other phylogenetic trees you've seen, but I think it illustrates this point very well. So these are our 3 domains, right? Archaea, more closely related to eukaryotes, as I hope you remember. The root is somewhere in here, and here's bacteria. But what do you notice about the things near the root of this tree in terms of this color scheme?
Extreme temperature.
Yeah, so all of these organisms, and some of them have been isolated, right, are thermophiles. And so this gives you the sense that, well, if they're near the root of the tree, maybe they evolved first. And so this gives some evidence that life may have evolved as a thermophile initially. Now, there's a number of methodological concerns about this, and actually thermophiles may evolve differently because they're at higher temperatures. And so there's some argument that this is artifactual, meaning it's not real biologically. That said, others have developed corrections for that, and they still place these thermophiles near the root of the tree of life. So that's kind of an interesting thing. So one of the premier researchers on thermophily, Carl Stetter, who was in Germany and has more recently been in San Diego, he may be retired at this point, but he studied thermophiles, and he was the one who cultured the earliest organisms growing above 100 degrees Celsius. So how do you think he did that? How did he grow something over 100 degrees, do you think? And this temperature of 121 or 122 should maybe sound familiar to you micro majors. If you've
Is it by
ever,
using an autoclave?
that's right. So what he did was he modified autoclaves, putting them under high temperature and high pressure, and he actually grew organisms in autoclaves. Of course, an autoclave is what we use to kill things, right? So this was pretty remarkable. So one of the things that's interesting to think about is, did life originate at hydrothermal vents? This would have the advantage of the high temperatures. It would also have the advantage of having lots of interesting chemistry that goes on there, right? Because things like hydrogen gas, hydrogen sulfide, methane get released. These can be used metabolically by modern microbes. And so they may have served as good electron donors. Okay. The other thing is hydrothermal vents are often found on the seafloor bottom, right? And being under all of that water actually protects organisms from the UV radiation that would have been coming in through the atmosphere, which was very thin at that point. It turns out that ultraviolet radiation is blocked very effectively by water. So if you're 2 kilometers down in the ocean, it's dark, right? So you couldn't be a phototroph, but you could potentially be a chemoautotroph living at a hydrothermal vent. There's also another school of thought that was put forward. Actually, this is a patent lawyer from Austria, Gerhard Wechterhauser, but he came up with an idea that life may have originated on clay particles, because it turns out clay particles are charged, and there's lots of interesting chemistry that can go on there, right? So if you're going to make metabolic reactions happen or chemistry work, before you have cells that can do this themselves with enzymes, et cetera, you want to find interesting chemistry to try and put forward as maybe the place where life, and that is what he came up with, but he never really followed that up. Now, this is taken from a general biology textbook. I hope most of you have heard of the RNA World Hypothesis. And so this is really centered around a main question of, we know that life uses RNA and DNA, right? But we use DNA as our main coding element, and we use RNA to do things, right? Makes the mRNA, that becomes a protein. Ribosomal RNA, that helps in translation, right? But it's possible that RNA was the first coding molecule, first macromolecule, and then evolutionarily later DNA became that. The advantage this has is that it turns out that there are RNAs that are self-replicating, and some of them are able to catalyze reactions in simple ways, kind of like an enzyme. So there's the postulate that's been forward that maybe RNA was the first coding sequence, or first coding macromolecule, and then DNA supplanted it later. So there's really two schools of thought. Did the chemistry come first, or did the cell come first? And we don't really know the answer to that. We know now modern cells get, packaged together. We know that we can take a simple lipid and we can get it to form a sphere without life, right? You can put it in water, and the hydrophobic nature of lipids causes them to ball up and form a sphere. And so that's like these lipoprotein vesicles here. How we get the cellular components and the coding molecules and the replicability is a huge leap, and we've never been able to really figure out which came first. But it's something interesting to think about. And so this is a fairly esoteric, but still an active area of research that people are looking at. So this is a study that's now a little dated, but it's from late 2009 or late 2000s. And they actually found that just with chemistry alone, using more plausible prebiotic conditions than the Miller-Urey folks did, they were able to generate ribonucleotides, right? Which then have this is more than just generating an amino acid. This is generating a potentially coding molecule in a test tube. All right. So one of the ways, even though we can't go back in a time machine and see what life is like billions of years ago, we can try to figure out what life was like. And we can use phylogeny, or the tree of life, to try to understand that. Kind of like I showed you with thermophiles, right? So trying to understand the history of life based on where we find organisms on the tree. And as was mentioned earlier, we talked about ribosomal RNA is kind of the gold standard. And this has been around since the late 70s, but really didn't come of age until the 80s and 90s. And so we use that as our standard gene for making the tree. And so I'll pose this question: why do we use ribosomal RNA as opposed to other genes? Does anybody know?
It's highly conserved.
Okay, it's highly conserved, so everybody has it. So eukaryotes have 18S, and archaea and bacteria have 16S. Okay, but that's a key thing. What else? Well, I'll give you some. So it's universally conserved. It does the same thing. Okay, so it's functionally homologous. Okay, it's alignable, and this is kind of a technical thing, but if we're going to look at gene sequences, we need to be able to line them up and say, oh, this is base number 10 of this species lined up with base number 10 of another species, right? For the 16S gene set. So that's important. And it's also slowly evolving and seems to be less prone to lateral gene transfer, that problem that I brought up earlier. So, and this is important because if this ribosomal RNA evolved 3.8 billion years ago, or around about that time, right? Then the modern organisms that are on the earth today have the same kinds of genes, but they will have mutated and mutated and mutated over and over and over. And so if we want to figure out who's really old versus who's evolved more recently, we need to try to avoid what's called the multiple hits problem. And that is, if you have a mutation in one base and then you mutate it again and you mutate it again, you don't know what the ancestral base was. It could have gone from an A to a C, back to an A to a T, right? So because the ribosomal RNA genes are essential, if you mutate them heavily, and they don't, usually mutations are negative, deleterious, bad, then that organism is going to die. So it turns out that the apparent rate of evolution of mutations is quite slow in these. And so that it made it a good target. And so we actually assume that it's not under heavy selection as well. Okay. Now I know this is, when you guys went through high school, you didn't really talk about kingdoms or anything, and you just talked about domains. But because I'm old, when I was in high school, we still talked about these things. It's important to note that it wasn't until the 80s and then 90s that textbooks started to get with the program. And really it was in like the late 90s, early 2000s that they got rid of these kingdoms. And we realized the importance of the work that Carl Woese at the University of Illinois and his researchers and his students and postdocs that went on to work in many labs all around the world. In fact, my postdoctoral mentor, Dave Stahl, was one of Carl's students. So he was he was early on in the system there. That really revolutionized our understanding of biology, which doesn't happen very often, right? So then we ended up with this 3-domain tree of life. Okay. All right. I think we are out of time almost. This table that I'm showing here basically just shows some different features that the 3 domains have. Okay. And so you can take a look at this. You don't have to memorize it, but it's interesting to see that there are some traits that are shared by bacteria and archaea, like the lack of organelles versus eukaryotes, right? And the 16S or the 70S ribosomes, which make the 16S, okay, versus the 80S. But then if we think about it, it turns out that bacteria and eukaryotes have similar lipids. They have ester-linked lipids as opposed to ether-linked lipids that archaea have. So you can see here, right? We have more in common with bacterial lipids than the archaea do, even though the tree shows that archaea are more closely related to eukaryotes. Cell walls are different across all three domains. Archaea and eukaryotes have 8 polypeptides of their RNA polymerases versus 4. So the take home from this is that evolution has taken a long time and it's messy. So it's not clear exactly how we got to where we are today, but it likely happened billions and billions of years ago. But there's been interesting evolution that's occurred so that some things have evolved differently in the across domains and some have remained the same. And so when we look at the tree of life, we need to think about what that means. And one of Carl Woese's sort of researchers that he helped train, Norman Pace, he really points out that we need to be very careful when we look at the tree of life because the evolution or these radiations for bacteria and eukaryotes and archaea are really poorly characterized. And so he argued you really can't look at the branching pattern here very effectively. Nonetheless, when we make trees, and this is from a general biology textbook, this is what we normally see with nice splits, okay? And this just highlights the fact that we have so few of the